All right, hello everybody. Today we're going to be talking about cloning and restriction enzymes. All right, so this is an organ and it's called the pancreas, and it produces something called insulin. Now, insulin is really important because it is a hormone that gathers sugar in your blood. And so we have this guy standing over here, and his name is Leon Noel. And so he's just walking around having a good day, and he's just looking around, looking at the sun and stuff like that. And then he realizes that he has a pancreas. But all of a sudden, he realizes that his pancreas doesn't produce any insulin. He has type 1 diabetes. So the question is, how can we help him? Because without insulin, he's gonna have a really rough time. Well, I just happen to know somebody that can probably help us. And it's this dude. And his name is
Bob.
Now, Bob also has a pancreas. And Bob's pancreas produces insulin. Now, what if we ask Bob kindly for some of his insulin, and then from his insulin, we can purify it and give it to Leon? Now, even though Leon's pancreas still doesn't work and it still doesn't produce any insulin, Leon can use Bob's insulin on a daily basis to regulate his blood sugar. But folks, there's a problem. Remember, Bob is not human. And so his insulin is a little bit different than human insulin. And so Leon over here, his immune system recognizes this. So it raises the alarm, and then Leon is having this allergic reaction. So what can we do for Leon now? He doesn't have any insulin. He needs it to survive. We have to come up with a solution. Well, the answer to that is somewhere you might not think of. It's in bacteria. Now, you might be asking yourself, why are we using bacteria? We've already used Bob, and because he wasn't human, we couldn't use his insulin. Now you're telling me that bacteria, which definitely are not human and don't even have a pancreas, are going to help Leon? And your answer would be yes. So bacteria have their own genome, but they also have these little circles inside of them. And these are called plasmids. And plasmids are these double-stranded pieces of DNA that can confer a lot of advantage for bacteria. For example, a bacteria like E. coli, which is quite common in your gut, can acquire one of these plasmids and become pathogenic, and then cause something like hamburger disease, which is an E. coli with an extra plasmid. So how is this going to help Leon? The central dogma of biology tells us that we go from DNA, which is like the blueprint for everything that we have, into mRNA, which is the print of that blueprint, into protein, which is the functional unit of everything that we do and everything that's in our body. And that is the precise mechanism that turns this friendly E. coli into E. coli Pro. The virulence gene on the plasmid, which is in DNA form, is being transformed into proteins which cause disease. Now, understanding that our friendly E. coli, we'll call her Anna, has the ability to take in foreign DNA with extra genes and express it into protein, is that we can maybe do the same thing with human genes. we can take a plasmid, put in a human insulin gene into our plasmid, and then insert it into a bacteria to get human insulin protein. Which means that our friend Leon over here, which was having a tough time with Bob's pig insulin, can use our human insulin without having an allergic reaction. Operation Save Leon. Okay, now that we have a plan, we need to make a to-do list. First, we have to find the human insulin gene. After we've done that, we want to insert it into our plasmid. After we have our special plasmid with the insulin gene, we want to express it, which means that we want to make it into a protein that we can use. And at the end, we want to give it to Leon. All right, so the human genome has 23 chromosomes, which are in pairs, so we have 46 in total. And the insulin gene is on the 11th chromosome. Now, let's say we sequence our chromosomes and we find the insulin gene and its sequence. And with this, we have our first step done. So now that we have our sequence, we want to insert it into a plasmid. But the question is, how can we do this? Because even though we have the sequence, we have to insert it into a completely different piece of DNA. Well, not surprisingly, the answer is bacteria. Specifically, one part of bacteria that are called restriction enzymes. Now, these restriction enzymes, which are also called endonucleases, cut through DNA. And they happen to be really useful in bacteria to protect them against this thing, which is called a bacteriophage. It's a virus that infects bacteria. So we have Anne over here, and all of a sudden she gets attacked by these bacteriophages. And so in this process, these viruses inject their DNA into the bacteria. But there's a catch. Our bacteria, Anna, has a restriction enzyme. And this restriction enzyme is going to cut through the DNA and protect Anna against this infection. But how does the story of Anna versus the bacteriophage relate to Leon? Well, these restriction enzymes can actually be quite specific. And they can cut through DNA and make sticky ends which we can use to attach other pieces of DNA in. But the question is, how do these restriction enzymes find where to cut? Well, your answer is something called the palindromic sequence. And in order to understand what that is, we have to go way back. If you pay close attention, you'd find out that there is something in common with all of these characters. And it has something to do with their names. All of their names are read both forwards and backwards, which is what a palindromic sequence really is. It's any sequence, whether it be letters like Leon Noel or DNA, that can be read both forwards and backwards. So let's start with Leon Noel. If you write the name backwards and draw a line in between the first and the last names, you will see that it doesn't matter how you read the letters, whether it will be from the top left or the bottom right, they match. And you could also do it from the top right and the bottom left. Now, all that this means is that Leon Noel is palindromic certified. Now, why am I going through this process to show that this name is palindromic? And how does this relate to our DNA? Well, what you have to realize is that your DNA is also double-stranded and is also anti-parallel, which means that one of the strands runs from the 5' to the 3', while the other one runs from the 3' to the 5'. All that really means is that they run in opposite direction. Now let's see if this sequence is indeed palindromic. We're going to start by a line that splits the sequence in half. Now we go through the same process and we look at the letters starting from the top left. Now looking at these letters, we can see that there is 4 adenosine residues on the top left. But if we look on the bottom right, we can see that the first letter starts with a T. That means that this is not a palindromic sequence. In order for them to be palindromic sequences, the letters have to match starting from the 5' end or the 3' end of either strand. But wait, what if we start counting from the second letter? We see that there is 3 adenosine residues starting from the top left and also 3 adenosine residues starting from the bottom right. And you guessed it, this means that this sequence is palindromic certified. Now you can really do this with any sequence. Let's say I give you this sequence and ask you whether it's a palindromic sequence or not. You first have to draw in the complementary basis to what I've given you. Then you can draw a line in the middle of the sequence if it's even, and then add in the 5 and 3' notation. What you might notice is that the 5' is on the top left and the 3' is on the top right. That's usually how the notation goes. The top starts from the 5' on the left, while the bottom starts from the 5' on the right. And remember, the reason for this is because the strands of a DNA sequence are anti-parallel. Now you can start counting from the top left and also the bottom right. And you can also do the same thing from the bottom left and the top right. You can see this sequence is palindromic certified. Now obviously in a test you don't have this much time, but this process will really put it into context for you and you'll get much faster as you keep doing it. Alright, so now that we know how to determine a palindromic sequence, we still want to help Leon. And we want to use this new information to insert our insulin into a plasmid. So here's our plasmid, but we want it to have a palindromic sequence so that we can use our enzyme to cut it and produce some sticky ends. And these sticky ends are going to help us insert the insulin gene into our plasmid. But for that to happen, we need to isolate our insulin gene, which we did before, and then find that same sequence both upstream and downstream of the insulin. By doing this, we're going to isolate our insulin with the same sticky ends that's going to allow us to stick them inside the plasmid. So now that we have isolated our insulin, all we need to do is to insert it into our plasmid using the enzyme DNA ligase. And after inserting our insulin into our plasmid, we're done with the second step of our to-do list. Now that we have our modified plasmid, we want to express it to get our human insulin. So initially we had our unmodified plasmid. We used our restriction enzymes to add in our insulin. And now we want some extra things in our plasmid in order to express it. And those are the origin of replication, which allows the bacteria to actually recognize our plasmid and in order to translate it into protein. And the second one is our antibiotic resistance gene. With our plasmid, we can insert it into bacteria, which could be E. coli, and then culture them. Now we can use our antibiotic resistance gene to select for the bacteria that have actually uptaken the plasmid. So how that works is that we have our bacterial culture, which has millions of bacteria. Then we use a bit of antibiotic and we put it into our culture. And what that does is that it selects for the bacteria that have the antibiotic resistance gene, and specifically the ones that are expressing the antibiotic resistance genes into protein. Because the ones that are not expressing the antibiotic resistance gene are prone to be killed by the antibiotic. So now we have a culture which only expresses the antibiotic resistance genes, which is a proxy that means that it's also expressing the insulin gene. So now all we need to do is to purify some of the insulin that's in our culture and then put it in a bottle to give to Leon. So it seems like we've gotten the results that we were looking for. And Operation Save Leon has been a success.